Hello, my name is Nathaniel Williams, and I'm a graduate student at Carnegie Mellon University in the Department of Biological Sciences and the Center for the Neural Basis of Cognition. I work in Carl Olson's lab, recording neural activity from the visual cortex of awake behaving monkeys. My talk today is about repetition suppression in low and high order regions of the primate visual cortex. Object recognition depends on multiple brain areas at different levels of the ventral stream hierarchy, especially the high-level region inferotemporal cortex, or IT, which receives visual input from the low-order region V2. And IT also has axonal projections which reach back to V2, although the function of these projections is not well understood. In addition to being sensitive to the identity of visual objects, IT is also sensitive to object recency, that is, how recently an object has been seen when it is seen again. One of the most robust ways that neurons in IT are sensitive to recency is the phenomena of repetition suppression, whereby when an image is presented first as a prime and then again as a probe, the response to the neurons in the form of action potentials or spikes per second is reduced for the second presentation compared to the first. This phenomena is observable in humans at the level of the hemodynamic response using functional magnetic resonance imaging. This figure from Cortex and Canwisher shows the hemodynamic response to the same image repeated, the red and the orange, is lower than to different shapes, the green and the blue. Highlighted areas of cortex, which show the strongest repetition suppression, are in inferotemporal cortex. The observation that suppression occurs when the same shape is repeated, but not when another similar but different shape appears, suggests that repetition suppression in IT is stimulus-specific. In addition to the observation that repetition suppression in IT is stimulus-specific, Devane and Vogels found in single neurons in macaque IT that there is some suppression that occurs even when the repeated stimulus appears in a different location, above rather than below fixation, and that's the solid versus the dashed and the gray lines. In addition to these changes in the neural response that occur during passive viewing, there's also a potential link to behavior in the form of an association between neural repetition suppression and repetition priming of reaction time. Buckner et al. Showed that subjects performing a move versus does not move categorization task, they observed both priming and repetition suppression under the same conditions. And despite what we know about repetition suppression, how does the visual system generate repetition suppression? With regards to network-level accounts of transmission between high-order and low-order regions, there are three main ways in which this could occur. Via bottom-up transmission from low-order to high-order regions, top-down from high-order to low-order regions, or independently in high and low-order regions. To investigate these three accounts of how the visual system generates repetition suppression, we recorded from neurons in both high-order and low-order regions of visual cortex, while macaque monkeys viewed sequences of images. To do this, we trained monkeys to fixate on a display screen in exchange for a juice reward. An eye tracking system monitored gaze, and when fixation on a central dot was attained, the trial began. If fixation was maintained for the duration of the trial, during which images were presented in sequence, a juice reward was delivered. During the experiment, an electrode in visual cortex recorded extracellular voltage deflections generated by neuronal action potentials, and the waveforms were digitized and then stored. The sequence within a trial was a fixation period of 300 milliseconds, a 300 millisecond prime presentation, which could appear in either the upper or lower quadrants of the screen, a 300 millisecond interstimulus fixation period, followed by a probe presentation for 300 milliseconds, in which it could match the prime in either location, identity, both or neither attribute, followed by a post-stimulus fixation, and finally by a juice reward. And what I'm showing here are some examples of stimuli used for the experiment when recording from both high and low order areas, and these stimuli were full-color background-free images of real-world naturalistic objects subtending 4 degrees of visual angle. In this experiment, there are 4 different conditions of interest which will be used as the basis of comparison to determine how repetition suppression is generated in high and low order regions of visual cortex. There are a both match condition, where the probe matches the prime both in location and identity, a loc match condition, where the location matches the prime but identity does not, an ID match condition, where identity matches the prime but location does not, and a no match condition, where neither identity nor location matches the prime. If we look at these 4 conditions as a 2x2 diagram of location match versus identity match, we can consider strength of suppression under each condition, represented as the size of the disks. We will then be able to observe and compare patterns of suppression in both high order and low order regions and submit these data to a 2-way ANOVA, where we can assess the significance of a main effective ID, a main effective location, as well as any interaction effects. And before I go over the results of the experiment, I'd like to talk a little bit more about the response properties of IT and V2 neurons and how they can be used to make predictions of the patterns of repetition suppression effects we might expect to see given the 3 different network level accounts of how suppression is generated at different levels within the ventral stream hierarchy. Neurons in IT have comparatively large receptive fields. In this schematic, the X's denote the possible stimulus locations, the cross in the center represents the fixation point, and the gray semicircle is the approximate receptive field of an IT neuron. IT neurons are highly selective for complex naturalistic images, so for this set of 6 images, a given IT neuron might respond strongly to only one of them. In contrast to IT neurons, neurons in V2 have comparatively small receptive fields. In this schematic, the gray circle is the approximate receptive field of a V2 neuron. Also in contrast to IT neurons, V2 neurons are not highly selective for complex naturalistic images and instead respond more broadly. So for this set of 6 images, a given V2 neuron might respond strongly to several images with no strong similarity to each other. This is likely due to V2 neurons being most strongly tuned to low-level stimulus features such as color, orientation, and spatial frequency, and multiple different objects may contain these preferred features. Based on what we know about the response properties of IT and V2, we can revisit these 3 accounts of how suppression is generated and make predictions about the patterns of suppression we would expect to see based on each account. For a bottom-up account of suppression in IT, we would expect to see that when the identity of the stimulus changes, there is a lack of suppression in IT, as well as a lack of suppression in V2, suggesting that the stimulus specificity of suppression present in IT is inherited from lower-order regions such as V2. For a top-down account of repetition suppression in V2, we would expect that when location changes but identity is the same, there is still repetition suppression in V2, suggesting that the location generalization of suppression present in IT, likely due in part to the large receptive field sizes, is transmitted to V2 from IT in a top-down manner. We would not expect this to be the case in a purely bottom-up account, due to the small receptive field sizes of V2 neurons, which are confined to a single quadrant of visual space. For an independently generated account of repetition suppression in IT and V2, we could allow for repetition suppression effects observed in V2 and IT to differ from one another and to fall more in line with the response properties of the respective areas. What I'm showing here is a peristimulus time histogram of the average response of 108 recorded IT neurons during all trials in each of the 4 conditions, plotted as firing rate and spikes per second over time. The no match condition is the dashed line, the loc match condition is in red, the ID match condition is in green, and the both match condition is in blue. The colored bars at the bottom of the histogram indicate times when each condition was significantly different from the no match condition by a cluster-based permutation test. We can see that in IT, using the no match condition as a baseline, there is no significant suppression in the loc match condition. However, there is significant suppression in the ID match condition, as well as greater suppression in the both match condition. Going back to the 2x2 diagram, I've schematized the results from IT, with the size of the disk representing the strength of suppression measured as the difference in firing rate between the prime and the probe 100 to 250 milliseconds post-stimulus onset. I then took the mean normalized strength of suppression and subjected the data to a two-way analysis of variance, which revealed a significant main effect of identity but not location, qualified by a significant interaction effect, whereby the strength of suppression in the both match condition was significantly stronger than that combined effects of identity alone and location alone. The pairwise comparisons of interest based on our predictions were whether or not there was significant suppression when only one feature, either identity or location, was a match to the prime versus the no match condition. A t-test comparing strength of suppression in no match to ID match revealed a significant effect of ID match alone. However, a t-test of the no match suppression to loc match suppression failed to reveal a significant effect. This confirms that the previous observations in IT, that repetition suppression generalizes across spatial locations but not across object identities, holds true under the conditions of our experiment. In addition to confirming these observations, we also observed that there is a synergistic effect whereby suppression is stronger in IT when both location and identity match the prime, even though the effect of location match alone is not significant. Now that we have confirmed the pattern of repetition suppression in IT, we can ask, what happens in the lower order region, V2? What I'm showing here is a peristimulus time histogram of the average response of 55 recorded V2 neurons. The experimental conditions were the same as those used for IT recordings, and the conventions are the same as those used for the IT data. In V2, note that there is no response to the prime in the no match and ID match conditions. This is because in these conditions, the prime was necessarily presented outside of the classical receptive field of the neuron being recorded from, due to the small receptive fields of V2 neurons. And this result was expected, therefore we only use the prime response in the loc match and both match conditions when calculating strength of suppression. When we consider the probe responses, again using the no match condition as a baseline, unlike in IT, there is no significant suppression in the ID match condition. However, also unlike in IT, there is significant suppression in the loc match condition, as well as greater suppression in the both match condition. Following the conventions from IT, I have schematized the results from V2 with the size of the disk representing strength of suppression. When the V2 data was subjected to a two-way analysis of variance, there was a significant main effect of identity, a significant main effect of location, and a significant interaction effect, whereby the strength of suppression in the both match condition was significantly stronger than the combined effects of identity alone and location alone. The pairwise comparisons of interest based on our predictions were the same as those in IT. A t-test comparing strength of suppression in no match to ID match failed to reveal a significant effect. However, a t-test of the no match suppression to loc match suppression did reveal a significant effect. When we consider the two conditions in which either location alone or identity alone changes, in contrast to what we observe in IT, in V2, repetition suppression generalizes across object identities, but not across spatial locations. Now that we have observed the pattern of suppression in both the high order and low order region of the ventral stream, we can again return to the predictions made by each of the network level accounts of how repetition suppression is generated in the ventral stream. A bottom-up account of transmission of suppression from V2 to IT predicts that effects observed in V2 would also manifest in IT. However, this is not what we observed. The repetition suppression we observed for different objects presented at the same location in V2 was not observed in IT, and so this casts doubt on a purely bottom-up account of repetition suppression. A top-down account of transmission of suppression from IT to V2 predicts that effects observed in IT would also manifest in V2. However, this is not what we observed. The repetition suppression we observed for the same objects presented in different locations in IT was not observed in V2, and so this casts doubt on a purely top-down account of repetition suppression. Instead, our data is most consistent with an independent account in which repetition suppression effects observed in IT do not transfer to V2, or vice versa. And with that, I'd like to thank my lab, the members of the Cone Lab I share space with, the NIH for funding this research, and thank you for your attention. Please contact me via email at npwillia@andrew.cmu.edu or online if you'd like to talk more about my research. Thank you.